And so now it's my great honor to introduce our second speaker, Melica Brodeur, who is an assistant professor in the Department of Obstetrics and Gynecology and the Department of Oncology at McGill University. She's an attending gynecological oncologist and a principal investigator at the Jewish General Hospital and the Lady Davis Institute for Medical Research, where she practices as a clinician scientist. She completed her medical degree at McGill, following which she undertook her residency at the University of British Columbia, pursuing a subspecialty and she pursued a subspecialty in gynecological oncology at the Université de Montréal. And then she went on to do a postdoctoral research fellowship at Memorial Sloan Kettering Cancer Center under the directions of Drs. Brita Wigaltz and Dimitri Zamarin, before starting her independent position here at the Lady Davis Institute in McGill. And her research focuses on understanding treatment response in gynecological cancers and discovering new therapeutic targets and treatment strategies to overcome therapy failure. She co-leads the Gynecologic Oncology Research Laboratory at the Lady Davis Institute and is co-director of the Gynecologic Tumor Biobank. Today, she's going to be telling us about modulating chromatin remodeling as a strategy to reverse immune invasion in the ovarian tumor microenvironment. And I'm very excited to hear her talk. So please join me in giving a warm, albeit virtual, welcome to our second speaker today.
Thanks so much, Ben. Can you see my slides and can you hear me well?
Yes.
Perfect. So thanks so much for the invitation and thanks to the audience for your attention. The goal today is to talk a little bit about reprogramming the immune tumor microenvironment using epigenetic therapy in ovarian cancer. And so depending on who's in the audience, you may or may not be familiar with the fact that ovarian cancer is not a single disease entity. There are different types, with the most common type being epithelial ovarian cancer that's divided in 5 main subtypes. And I've listed here as well in the black box a few other less common types of ovarian cancer that are not quite epithelial ovarian cancers. The cancer statistics, unfortunately, still show that ovarian cancer is a deadly disease. Most patients present in advanced stages, and despite the known heterogeneity of this disease, we're still giving a one-size-fits-all treatment with a first-line cytotoxic chemotherapy. And so, unfortunately, the 5-year survival remains very low for this population at 30%. And so we definitely need to do better for our ovarian cancer patients. Meanwhile, immune-targeted therapies such as immune checkpoint blockade have shown amazing success in multiple cancer types, including some gynecologic cancers like endometrial and cervical cancers. But unfortunately, till now, immune checkpoint blockade trials in patients with ovarian cancer have shown really disappointing results that you can see here from the graphs, with poor response rates and really no clear biomarkers for response. What was interesting, however, from these studies was that there were a subset of ovarian cancers called clear cell ovarian cancers that had higher response rates, and it might make sense a little bit later on in this talk why that might be so. As well, epigenetic therapies, including targeting chromatin regulatory factors or their downstream pathways, have emerged as promising strategies in recent years. And so the switch-SNF complex plays an important role in gene regulation, including oncogenes, tumor suppressor genes. And I kind of like to compare it usually to an on-off switch of gene transcription. As many of you may or may not know, the complex is made up of multiple subunits, and 29 genes encode for these different components. SMARCA2 and SMARCA4, also known as BRM and BRG1, are 2 critical ATPase subunits that utilize energy from ATP hydrolysis to mobilize the nucleosome and remodel chromatin, so essentially making chromatin more or less accessible to transcription. And SMARCA4 will be the subunit that will be highlighted throughout the talk today. Several studies had shown that a subset of ovarian cancers is characterized by an immunogenic tumor microenvironment that can make them more sensitive potentially to immune checkpoint blockade. And so before I joined the Sloan Kettering team in New York, our research group had characterized the immune tumor microenvironment of a series of patients with SMARCA4 mutated ovarian cancers called SCOOTs. And in the study, it was found that there was a prominent T cell infiltration in the tumor microenvironment, and the tumor-associated macrophages were highly expressing PD-L1. And also mentioned in the same paper, anecdotally, some of these patients did respond to immunotherapy, whereas they hadn't responded at all to standard of care chemotherapy. So this is interesting in the context of tumors that are actually known to have very low tumor mutational burden. And typically, tumor mutational burden is a biomarker for more immunogenic tumors or tumors that would be more responsive to immunotherapy. So then we decided to query the TCGA, and we found that actually low expressing SMARCA4 ovarian cancers have upregulated pro-inflammatory immune cell population infiltration. So you can see here leukocytes, Th1 cells, and M1 macrophages, which are all more pro-inflammatory. And so with this in mind, we wanted to understand the immune tumor microenvironment interactions and mechanisms of increased immunogenicity of SMARCA4 altered ovarian cancers that could imply a potential role for targeted therapy in ovarian cancer. So to do this, we had to develop SMARCA4 deficient human and murine ovarian cancer models with their respective controls to be able to answer the research question. So we used two murine ovarian cancer models, so ID8, which is a TP53 wild type murine cancer, and UPK10, which is a TP53 mutated murine ovarian cancer. And given that we'd be conducting a lot of immune tumor microenvironment studies, most of the in vitro and in vivo studies were mostly focused with the murine models that we developed. And then we did in vitro validation using the human OAW28 model that is a TP53 mutated ovarian cancer model. And so from the murine models, we derived clonal cell lines with which we transfected either with single guide RNA targeting SMARCA4 or non-target control using CRISPR-Cas9 technology. So once our models were ready, we performed RNA sequencing, comparing expression changes with or without SMARCA4 deficiency. And what we found was that downstream immune-related genes and pathways were upregulated, including important interferon-stimulated genes, chemokines, and antigen presentation genes. And so with this data, we then focused on these specific downstream immune pathways. So in particular, here on the left, you see the type 1 interferon genes that were significantly upregulated with a list of these key genes in the pathway. We then were able to further support our RNA sequencing analysis with flow cytometry data. So we found here on the right panel a significant increase in MHC1 expression here in blue on the surface of SMARCA4 knockout cells compared to the control, suggesting increased antigen presentation ability of tumor cells with loss of function of SMARCA4. And we saw similarly the same difference when we added interferon stimulation. So given SMARCA4's role in the chromatin remodeling complex, we next wanted to understand whether the loss of SMARCA4 led to changes in chromatin accessibility that would explain some of the immune-related changes that we observed. So we performed attack sequencing, and amongst the genes that demonstrated increased chromatin accessibility, interferon and chemokine signaling pathways were upregulated. And many of the genes that had increased expression in our RNA-seq analysis also had increased accessibility on attack sequencing. So here are two examples with CXCL10 and CCL2, and you can see here in red, the SMARCA4 deficient model has a clear increased peak at the promoter site compared to the non-target control. And so We then decided to analyze our data further to determine if any recurring motifs were enriched in the open chromosomal regions affected by SMARCA4 deficiency. And we identified 3 significantly enriched motifs that are bound by transcription factors that regulate immune genes, which include AP1, NF-kappa beta, and IRF, so interferon regulatory factor. And that's the one I'll be focusing on mostly for the remainder of the talk. So with these data so far, we wanted to further explore interferon-stimulated gene upregulation mediated by the type 1 interferon signaling pathway. So for those of you that may or may not be familiar with this pathway, ISG transcription is initiated when the type 1 interferon receptor is stimulated by the secretion of interferon alpha or beta. And so interferon alpha beta is produced in the nucleus after key upstream regulators such as IRFs get activated by any kind of stimulus, which could be from different triggering events such as DNA damage, increased double-strand RNA or retroelements. And so our aim was to understand and test the key players in this pathway that led to interferon-stimulated gene induction. So we started with blocking the type 1 interferon receptor, since that seemed like the most logical explanation. So we performed these blocking assays, but what we noticed was that it actually did not attenuate the ISG expression in our SMARCA4 knockout cells. So on the left panel here, the SMARCA4 wild type cells are in black. And in the colored bars, these are the SMARCA4 knockout clones treated with or without the receptor blocker. So in the dark is without, and in the lighter color, it's with the blocker. So you can see that there was no difference, no statistical difference with or without interferon receptor blockade. So this suggested that transcription activation of interferon-stimulated genes are not fully dependent on the activation of the type 1 interferon receptor by secreted interferon alpha or beta. And so given these data, we then decided to evaluate whether key upstream regulators, such as the IRFs, were directly responsible for activation of ISGs. So what we found is that both at the RNA level and at the protein level, IRF3 expression was upregulated in our SMARCA4 knockout cells compared to control. And not only that, but we also noticed that there was an increase in the activated IRF3, and we measured this by looking at the nuclear to cytoplasmic ratio of IRF3 in the context of loss of function of SMARCA4. And here's a positive control using poly-IC. So to link IRF3 induction with ISG upregulation, we performed knockdown experiments using shRNA targeting IRF3 or non-target control in the SMARCA4 knockout cells. So what we saw was that knocking down IRF3 abrogated the ISG upregulation, reinforcing the direct link between IRF and ISG. So we see here in the panel that the black bar represents the SMARCA4 wild type cells, and in the blue and orange, these represents the SMARCA4 knockout cells that are transfected either with an SHNTC in blue or with the SHIRF in orange. And you can see this clear abrogation of these ISGs. So with these findings, we decided to examine the IRF3 binding sites at these interferon-stimulated gene promoters as a potential mechanism that could explain the interferon alpha receptor independent ISG upregulation that we observed with SMARCA4 loss. So using our ATAC-seq data, we found that IRF3 motifs are enriched at the transcription start sites of different ISG locus. So here's an example looking at OASL2. And this suggests that IRF3 may bind some sort of interferon-stimulated response element that directly triggers ISG expression. We also looked at the publicly available data sets from CHIP Atlas, showing IRF3 binding sites at the promoters of key ISGs and human cancer cell lines. So here we have an example of a multiple myeloma cell line, and as well key binding at promoters of interferon-stimulated genes in murine immune cell lines, such as macrophages, for example. So knowing that ISG upregulation was not dependent on the type 1 interferon receptor and seemed to be directly affected by IRF3 activation, we then focused on potential upstream signaling mechanisms leading to IRF3 activation. So based on previously published data on in the literature, we hypothesized that chromatin remodeling associated with loss of SMARCA4 could lead to re-expression of non-coding and transposable elements, which carry potential to engage innate receptors that have been shown to lead to type 1 interferon pathway activation. And so we first analyzed our RNA-seq data looking at non-coding genes. And what we found is that the loss of SMARCA4 in the murine ID8 cells induced a significant increase in a type of double-strand RNA or transposable element called long terminal repeats, which include endogenous retroviruses. And we confirmed this as well by qRT-PCR in our UPK10 model as well, using a panel of endogenous retroviruses. And so with these findings, we wanted to determine whether the RNA sensing pathway could be potentially driving the ISG upregulation, possibly through detection of increased long terminal repeats that are being expressed in SMARCA4 knockout cells. So we performed shRNA knockdown in the mitochondrial antiviral signaling protein called MAVS. It's a key downstream adapter and RNA sensor mediating both RIG-I and MDA5 signaling. And so as you can see here from the right panel, knockdown of MAVS versus non-target control in our SMARCA4 knockout cells resulted in abrogation of expression of different ISGs. And so you can see here in black the SMARCA4 wild type cells, and then in the colored bars, these are the SMARCA4 knockout cells. In orange, the cells were transfected with non-target control versus the green and blue were transfected with the SH MAVs. And you can see this clear abrogation across different ISGs. So with these in vitro data, of course, we wanted to see if this increase in immunogenicity was going to be observed in the tumor microenvironment. So we inoculated immunocompetent mice intraperitoneally with SMARCA4 knockout versus non-target control cells and collected ascites after 3 weeks. And then we performed spectral flow cytometry to examine a panel of both innate and adaptive immune markers. And so we were able to get many results, but I'm highlighting a few for you here. So in a nutshell, we noticed that our CD4 and CD8 T cells were significantly more activated in our SMARCA4 knockout tumors, as well as evidenced by the PD1, the higher PD1 expression in these knockout tumors. We also noticed in our natural killer cells that they were significantly more activated, as you can see here by the increase in the granzyme B expression in our SMARCA4 knockout tumors. We also chose to examine the myeloid compartment of the tumor, and we saw really interesting changes in the microenvironment. So there were more dendritic cells that were present in the tumor microenvironment, and the dendritic cells expressed more MHC2, suggesting they were more capable of antigen presentation. We also imaged our mice and noticed that the tumor velocity was significantly impaired in the SMARCA4 knockout tumors, suggesting an immune anti-tumor suppressive effect. So of course, naturally, we then wanted to see if we could therapeutically target SMARCA4 and recapitulate these results. So we inoculated mice with ID8 parental cells and treated them with a commercially available BRM BRG1 inhibitor versus vehicle. And as you can see here on the right panel, both the CD4 and the CD8 cells were significantly more activated. You can see here increased expression of ICOS, PD1, and granzyme B in the SMARCA4 treated group compared to vehicle. And so equally wanted to look at the myeloid compartment, and what we found was that the dendritic cells were again more prominent in the tumor microenvironment, and they appeared to again be more antigen presenting with the increase in MHC2 expression. We looked at the growth velocity again of these tumors and found that the treated tumors had a suppressive growth effect by imaging, as well as looking at weight measurements for ascites accumulation over time. So with these results, we were able to conclude that the disruption of the catalytic subunit of the switch-SNF complex led to increased tumor immunogenicity, and that this was likely not dependent on the type 1 interferon pathway and more mediated through IR3 activation. And so this, the loss of function of SMARCA4 leads to a more primed, if you want, for lack of a better word, primed tumor microenvironment to sensitize potentially these tumors to immune-targeted therapies. And so at the moment in our lab, we're conducting some studies where we're combining BRG1, BRM inhibition with another epigenetic therapy to try and sensitize the tumor microenvironment when we combine with immune checkpoint blockade. And so with that, I want to thank the people in my lab and in the core facilities that have tremendously helped with this project, as well as my mentors at Seoul Kettering and the scientific input I've gone from mentors in the Chapinelli lab and the Van der Heyden lab. And of course, we can't do any of this work without our funding agencies. So thanks to them as well for supporting this work. And I'll take any questions if there are any. Thank you.
Awesome. Thank you. That was a wonderful talk, a really cool project. So for everyone in the audience, you can type your questions into the Q&A box and I'll read them out, or you can click the raise hand button and I'll unmute you to ask the question yourself. But I have a bunch of questions, so we'll start with those. So in your SMARCA4 knockouts, presumably SMARCA2, its paralog, is partially compensating for loss of SMARCA4. So what I'm wondering is, do you think the effect you're seeing is due to kind of a general perturbation of the switch-SNF complex, so kind of lowering the activities of all switch-SNF? Or is this somehow specific to SMARCA4 versus SMARCA2 in terms of the regulation of the LTRs and this immune stimulation?
Yeah, it's a great question. Given that for the knockout models, obviously we knocked out just SMARCA4. So it is possible in that case that SMARCA2 could have had some compensation there. But given that the therapeutic targeting the studies that we did was using an inhibitor for both BRM and BRG1, I suspect that any role that SMARCA2 or BRM could have played may be minimal, given that we could see the same effects when we inhibited both. But for sure, I can't say that for sure, but I suspect that based on the data that we have for this project.
Yeah. And do you know in your cancer models, is SMARCA4 or BRG1, is it more highly expressed or the more abundant one than BRM?
Great question. We didn't actually look at necessarily the baseline expression of BRM and to see how that changes when we change the expression of SMARCA4. But it's definitely something to look at, I think. I think mechanistically it could potentially help explain what we're seeing, but I didn't do that, no.
And then for the inhibitor that you have, what in the SMARCA4, SMARCA2 is it targeting, and how is it working?
Good question. This inhibitor is one that is one of the more commonly used inhibitors in the literature right now. Now, exactly what it inhibits and how, I couldn't tell you. I wouldn't be able to say.
And then when you're like, you have quite nice results in the animal model with the inhibitor. Um, you know, what obviously switch SNF is important in lots of cells, and immune and some immune cells especially. So like, how are you approaching, like, when you treat with the inhibitor, it's going to affect your cancer cells as well as your immune cells. And so is it a bit of like a balancing act for dosage to find the right amount of the inhibitor to get your cancer cells versus your immune cells, or how do you approach that problem, I guess?
That's actually a great question. And I did initially have to do some troubleshooting. So the dosage that we used was initially based on published results when with this inhibitor that was developed. Um, and we had to work a little bit on the optimizing the schedule, because it is true that the effect of the inhibitor will be sensed by both the tumor and the immune cells. And what we noticed was that if we harvested the tumors very close to sort of the last treatment with the inhibitor, the immune cells actually also got affected negatively. And so we had to optimize it by actually giving sort of a drug holiday. So giving the treatment 4 days on, 3 days off for 2 weeks, and then allowing a bit of time before harvest. And that was actually based on publication from a few years ago, not with this inhibitor, but with another targeted therapy that had showed the same thing, that the way that the drug is scheduled and the timing of collection will greatly affect the results you see in the immune tumor microenvironment. So I think probably it's sort of a bigger picture comment, but I think this is potentially how good drugs are potentially tossed aside because of poor optimization of the route and administration and dosing of inhibitors. So, but that's a bigger problem than the scope of today's discussion.
And do you have to take similar considerations when you're thinking about dual treatments, like the combination with the DMT1 inhibitor?
Absolutely. So the nice thing was that because there's a good amount of literature on DNMT1 inhibitor, and one of my mentor, Kate Chapinelli, who works a lot in this space as well, has provided a lot of insight into this work. We did have to do a bit of dose optimizations at first, and even in our animal model, we had to test different doses to see how the combination would affect the mice in terms of toxicity. And indeed, it's good we did that, because the higher dose that had been used alone, so without combining with another drug, was tolerated in another study, but when combined with another epitherapy was quite toxic. So we had to decrease the dose, and we're waiting for results from that. So more to
Okay,
come.
there's a question from David Arnosti who says, do you expect that the inhibition of switch SNP would activate the RIG1 dependent IRF3 signaling in a cancer type specific manner, or do you expect that this response is common to most or all cancer cells?
Great question. We did do the same analysis using sort of targeted panel of endogenous retroviruses in a melanoma model, and we also saw increase in endogenous retroviruses when we knocked out SMARCA4 in a melanoma model. So I suspect it's not just for ovarian cancer, and it may be for other cancers as well. So.
And
Yeah.
do you think, do you remember, or do you know if it was the same LTRs, like the same families that were getting upregulated, or is it more kind of a general LTRs are going to get turned on, but in a given cell type, it might be different which actual ones are responding?
I actually think it changes. If yeah, I mean, you know, the in the melanoma model, we didn't do RNA sequencing, so I don't have as much of a full panel. But even in the panel of, if I'm not mistaken, about 8 genes that we looked at, endogenous retroviruses, there were differences in the melanoma, which ones were upregulated versus in the ovarian. So I do suspect that there are some differences. Now, does that matter in terms of the impact on, activating the type 1 interferon pathway? That I'm not sure.
And um, do you expect that the DMT inhibitor is also going to kind of work through the I'm a bit naive on what's known about the DMT inhibitor, but I assume LTRs are activated in that context as well. And do you think that's where you're going to kind of see the synergy? Is this kind of stronger activation of the immune um in this RIG1 dependent IRF3 signaling, or is there some other mechanism that may be happening in those?
Yeah, there's a beautiful paper in Cell. I believe it was published in 2015, and the first author is Kate Chapinelli, who's a PI now in Georgetown. And she published a beautiful paper going over DNMT inhibition and how it does activate endogenous, double-strand RNA sensing pathway, and which is why it was the logical choice to combine. It's a well studied class of medications, and it seems to also activate the type 1 interferon pathway similarly, and so it made sense to combine BRG1 inhibition with the DNMT inhibition. But now I know that the toxicities are additive, so there's that to consider in terms of dosing, and to check if a reduced dosing would give the same effect.
And then like thinking about this concept more broadly, like, SMARCA4 is obviously knocked out in, a decent number of cancers. Um, it's, a common common cancer mutation. Do you think that this concept could be applied to some of those cancers in terms of looking for immune activation in the SMARCA4 knockouts? And have people been looking at things like a DMT inhibitor in those contexts?
Yeah, so actually there's a more clinical paper that was published looking at non-small cell lung cancers that have the highest proportion of SMARCA4 mutated tumors. And actually what they showed was that in those that had the SMARCA4 mutation versus those that didn't, there was a higher response rate to immunotherapy, which sort of, suggests that potentially these tumors are more immunogenic. Um, I can't tell off the top of my head if studies were done using DNMT inhibition in lung cancer. I would imagine so, but I don't know off the top of my head.
Okay, and so we have a raised hand, so I'm gonna let Julietta talk to ask her question. Let's go ahead. Good. You can unmute yourself now and ask your question. Oh, okay. Or maybe we'll come back to you. There's a few other questions in the Q&A box. So let's move. So Genevieve has a question or a comment. So she's saying somewhat similar results with CDK9 inhibitors was shown to work through phosphorylation of SMARCA4, which actually activates SMARCA4 targeting. So anyway, she's got a comment saying there's some similarities to what's been seen with CDK9 inhibition that may be interesting to think about with how those inhibitors may be affecting SMARCA4. And there's
Oh,
also
thanks for the comment. I'll note it down.
yeah, and she's got a paper reference there to kind of follow up with.
Great, thank you.
And then there's a question on the attack seek. So are the upregulated genes with increased attack-seek signal in the SMARCA4 knockout occupied by SMARCA4 in the wild type condition, like if you've done CHIP for them, for SMARCA4, for
No,
example?
unfortunately, honestly, the reality is that there's so many questions and so many more experiments that honestly I would have loved to do, and at some point we had to kind of just draw the line. But there are so many unanswered questions and more to explore with this project in terms of mechanisms, and going even further, about even with the role of IRF3 in activating the ISGs. So it's a great point, and it's something definitely that if I if I could spend more time and more experiments would be something interesting, and it's something to consider for this sort of second part of the project when we're combining the dual therapy to look into. So thank you for that suggestion.
And then we've got kind of a broad question here at the end, which is, can you elaborate on the correlation of the immunogenicity and homeostasis in the tumor microenvironment?
Wow, that's very broad. Anything in particular about homeostasis? Like,
Yeah, great question. It
I'm sure it's a bit of like a read my mind kind of question. Um, if that person can elaborate
nor
a little bit more.
normal tissue versus tumor tissue. So they've added.
Okay, okay. So how it affects the tumor microenvironment versus normal tissue, basically, I guess. That's a great point because given that given that this inhibitor will affect all cells, unlike, for example, a class of medications that target specifically uh, receptors on cancer cells that are not present on normal cells, this is a great point and probably something to consider when we're harvesting to look at non-tumor tissue and to see if there are any changes in the treated versus untreated group. So that's a great point. Maybe it's something we'll answer in the second part of this project. Yeah, but great point.
Well, we're at 1 o'clock here on the East Coast, and we've had a good discussion. So I don't see any more questions. So we're going to wrap it up there. So just thank you everyone for attending. Thank you so much to both our speakers for a great session, and we'll be back in 2 weeks with our next FragileNucleosome seminar.